Okay. So today we are switching from the immune epitope database, which we talked about yesterday, to the analysis resource, which is essentially a companion website to the IEDB database. And I'll start with the acknowledgments, and we desperately need updated pictures before. There's obviously the IEDB team involved in this. Today you'll hear talks from Edita, Sinu, and Julia, who are here. And then Linus is performing some work on essentially the basic maintenance of the servers. And then we have lots of collaborations with the CBS and DTU groups in Denmark, and for example, visiting from the group right now. But Morten Nielsen, Ole Lund, and Søren Bruce have contributed a lot to this, the tools that you see as well. There's two main categories of tools in the Epitope database. One are prediction tools, and the other ones are analysis tools. So prediction tools are machine learning algorithms that take the data in the IEDB and try to find generalized patterns and use them to predict new similar data. So that specifically applies to MHC class 1 and class 2 binding data, MHC class 1 processing and immunogenicity predictions, detailed epitope predictions, and all of those are going to be explained in more detail. So again, like yesterday, I'm kind of giving the broad level overview, and then the other talks are going to dive into all of these in more detail. The other parts of tools that we have are the analysis tools. So analysis tools are essentially things where either you have your own epitope data set and want to analyze it, or maybe you query the IEDB for some data and then you want to analyze it further. So those are not for predicting epitopes, but they're using epitopes that you have from some source and getting more information about them. So epitope prediction tools. All of them are essentially, as I said, trying to generalize patterns of data that we have in the IEDB. And as an example, I'll talk about MHC class 1 binding prediction tools and a pretty simple algorithm that is easy to explain. So the experimental basis of the MHC class 1 binding predictions are binding assays. So in these assays, you have typically a ligand of interest that you measure for binding to a purified MHC molecule. And the vast majority of data we have measures IC50 values. So an IC50 value is an inhibitory concentration, 50%. You have a competitive ligand that binds very well to the MHC molecule. You're titrating in the peptide of interest and asking, when do I have inhibited 50% of the binding of the reference ligand that is labeled? And so essentially this is the concentration, and the lower this concentration is, the higher the affinity. You need to titrate a little bit, and you immediately have 50% drop of binding. Clearly, it's a high affinity group. And run under the right conditions, the log of the IC50 is, so this is actually, it comes to KD, and is approximately an approximation of binding free energy of this reaction. So you can say, well, great, you can do these binding assays and apply them to T-cell epitope predictions. By just going through and taking, say, all the open reading frames from the pathogen and just taking all the peptides, making them, and testing them in the binding assay. The problem is obviously that would cost a lot of money. Because making peptides costs a lot of money, and testing them for binding costs a lot of money. So rather than doing that, can we learn from a set of peptides that we have measured what other peptides will be binding as well? So, and that's what the predictions are for. And ultimately, when you do want to do these predictions, what you're looking for is some kind of function from some kind of function space, where you look for a function where, given the peptide sequence, it'll approximately give you back the affinity that the peptide had in the assay. And that's all what machine learning is. There's many different approaches for this, like neural networks, support vector machines, hidden Markov models, linear programming, etc. And what they differ in is, what do they mean by find? What kind of function space are they looking in? And what do they consider an approximation of this prediction to measured affinity? What do they think is successful? So I'll give what's probably the easiest to explain example for such a machine learning algorithm. So again, you have as an input data set peptides with measured affinities, and these are here expressed as log IC50 values. So the value of 1 would be 10 nanomolar. And you're trying to derive a matrix like this, where for every position in the 9-mer peptide, you get like a value that contributes to the affinity. And so say if you have that peptide sequence, you take the corresponding values from this matrix, you sum them up, you add an offset, and then you have your predicted log IC50. So machine learning for positional scanning scoring matrix like this just means, okay, if I have a bunch of, preferably a very long list of peptides with measured affinities, take a matrix like this and change these values in each position until I have a good approximation of the measured values when I do the summing up the matrix values. And that's the short summary. Then there's a bunch of little things one can do to make this better. But what I'm going to go next for is, once we have these methods, and as I said, there's many, many different ones, how do we evaluate, how do we say which one should be used? This is actually what I came here for as my postdoc. That was the one reason why I really wanted to come to Alex's lab, because I knew he had thousands and thousands of data points, and I always had like tens, not tens of thousands, like tens, and to go where the data is. And so we had data from Alex Sette and Søren Buse's lab, and it was massive. It was way bigger than anything that was out there. It's like 50,000 data points, and that was essentially as the IEDB got started, but that's what started off with. So we could take all these data points and run this like 48,000 total binding values. We could run them now through the available public prediction algorithms. And so at the time, the two most commonly used algorithms were SIFPIDE and BIMUS. And essentially, SIFPIDE gives you a prediction score ranging from 0 to something like 35, and BIMUS gives you some kind of predicted half-life. So none of these actually really predict IC50 values, which is one of the downsides. But you already see that there is clearly a correlation in that higher predicted scores here correlate with low IC50 values, so high affinity binding, and similar for BIMUS, long half-lives correlate with low IC50 values. So this is one way to compare these things and calculate R-squared values, but there's certain issues with the R-squared values, specifically because, as I said, these are not really, you're not predicting things on the same scale. So you don't really want to use an R-squared value, you want to use some non-parametric measure. One of those is the residual operating characteristic curves. So there we are trying to evaluate how good the scoring scheme is in dividing peptides into two sets, namely binders and non-binders. And then we're using the traditional cutoff of 500 nanomolar, which is for AO201, had been shown to be like 90% or so of all known epitopes have a affinity of 500 nanomolar or better. So we're trying to divide out here those peptides that are binders from those that are not. And you can say, okay, I can just take the score here and say, okay, this everything that is above this score is predicted to be a binder, so these guys, and everything below is predicted to be non-binder. And then I get true negative, false positive, false negative, and true positive predictions in my 2x2 table. And I can do this now for every prediction score, and I always get this 2x2 table, and can use this to then essentially plot as a function false positive rate and the true positive rate of the prediction. That is the receiver operating characteristic curve, and in an ideal case, it would go straight up and over there, so that there's no false positives and you're getting all your 100% of true positives. But this is what in reality it looks like, and here you can say, so BIMUS performs better in this case than SIFPIDE. Yeah. And both are extremely clearly better than random. So we could do this for a bunch of, this is weird, a bunch of publicly available methods at the time, and we ran all these data sets through, and so that was informative in the sense that this is how current tools perform, but it is not really a very good way to learn going forward which approach works better than the other, because there's two key influences here. One is how good is the prediction method that is used? So is the neural network working better than the matrix, say? But the other and actually dominating factor is what was the training data set available to the people who were making these methods? So essentially, we wanted to have another benchmark in which we don't have this problem that, I mean, all these things were from web servers and they were all generating completely different data sets. We wanted to use something where we have control over what's the training data set utilized, and we did this by using internal cross-validation. So these are methods that we had in-house at the time, and that we had complete control over what is used to train the method and what is used to test the method. So cross-validation works like this. You take the whole data set and split it in 5 parts in our case. You take one part, leave it out. You take the other 4 and train your matrix or neural network or whatever on those 4, and then ask that prediction method to predict the affinities of the peptides that were left out. So in the upcoming talks, we will show results, but this is essentially the 2 kinds of evaluations we're doing. So either a cross-validation on in-house data sets where we have complete control over the methods that we can generate new predictions for, or we go through external databases and externally available tools and try to evaluate their performance on some data set that isn't available to them yet. So there's going to be much more detailed information in the other parts, but essentially these approaches are used for B-cell epitopes. B-cell epitope predictions are tricky. The historic ones have been using amino acid-based scales, such as hydrophilicity scales. There are also machine learning predictions out there that have been getting better and better. These apply to all organisms. There's not thought to be a difference between antibody epitopes between, say, humans and mice or so. But overall, the prediction performance is rather poor. With AUC values, which is the area under the curve of this ROC plot that I just showed you, between 0.6 and 0.10. For MHC class 1 binding, we have a lot of data for different human alleles, non-human primates, non-primates. The accuracy is very high. These predictions work extremely well, with average prediction performance with AUC greater than 0.9. In general, these haven't been making leaps and bounds because we are close to the, actually not, and we made some comparisons and could show that the prediction methods at this point are just as accurate as the binding measurements. So when we have a big discrepancy between the prediction and the binding, quite often if we synthesize a new peptide and measure that again for affinity, in the next round it is more closer to what the prediction was. The typically peptide synthesis we do is we use, we have to, that's one of the big variables that is hard to control for. For class 2 binding, we cover humans and non-humans, although much less than for class 1. The accuracy is lower in comparison to class 1, but definitely higher than antibodies. And there have been substantial improvements in this area in the past few years. So we think now that in terms of the ability to predict peptide binding to MHC class 2, that seems to be working quite well. There are some problems in using that to predict epitopes, and Tina's going to talk about that. So then there's the processing prediction tools, and I'll talk about that later actually. So for MHC class 1, peptides that are actually recognized by T cells undergo proteasomal cleavage first and have transport. And these are independent mechanisms that select additional subsets of the peptides that can actually bind to MHC class 1. So that's one approach of actually modeling these separately. Another approach is to just take peptides that are eluted from MHC ligands and use them to directly train predictors. These has been only implemented for humans. We have done some work on mice. It hasn't been that successful because mice just seem to be less selective. And these do improve MHC class 1 binding predictions alone, but those improvements are rather small. One issue is that for MHC class 1 binding, we have much better data sets available, larger data sets, and we still haven't performed really a definite large-scale validation that compares how much is gained by these. So then for the analysis tools, I was going to, and actually, Ward, how much time do I have? Is it until half past or until 20 past? Yeah. Okay. Start a little bit. Yeah, because I'll be at half past. So for the analysis tools, again, those are the tools that can be used to add value to Epitope datasets that either you can get by querying the IEDB or that might be in your own lab. I'll give an example of conservation of swine flu, of how these we actually applied these kinds of tools in a real case scenario. So as you know, in spring 2009, there was this spread of swine flu coming from Mexico, and at the time, actually, there was lots of people dying, and they had like a expectations of really, really high mortality rates. And the sequencings became available in 2009. We thought, well, you're funded for these kinds of things, you should be doing something. So the question we asked is, and at the time, essentially people were saying this flu is so different from the other flus that are out there that there's essentially no immune protection to it. And that's why it causes this high mortality. So what we essentially asked, can we quantify that based on data that we have in the IEDB? And so what we did is we queried the immunonegative database for all epitopes that had been previously described to be contained in Influenza A viruses. And then we looked at what are the circulating influenza strains that have been around in the 20 previous years. Then we asked, okay, given that we know what parts of the sequence can be targeted by the immune system and what kind of proteins and peptides have actually been circulating in the past 20 years, can we ask which ones of these epitopes have been previously essentially stimulating people and are likely to cross-react with the current strain. Yeah, that's what I just said. And then we followed it up experimentally. So this is the old site. Was apparently lazy and didn't do an update, but the query is straightforward. Say for B-cell epitopes, you're looking for the source organism, the antigen being Influenza A virus. Then you ask for B-cell epitopes in this case, and we do the other ones as well. And you ask for that these are recognized by humans. Then you get a bunch of peptides as a result. You can take them, you can export them into Excel as you saw yesterday, and now you can plug them into the conservancy tool. So in this case, the conservancy tool takes a bunch of peptides as an input and a bunch of protein sequences. So here we put in the 2009 pandemic influenza strain, and then essentially ask, so which ones of these peptides are found in these 2009 strains? And then essentially you get this kind of output where actually we found you get, like, for each of the peptides, the degree of conservation of it, and essentially then that summarizes that we compared the 2009 pandemic strain, which at that point was called swine origin influenza virus, now it's typically 2009 H1N1 pandemic strain. And we compared that to the seasonal strain of the previous year that circulated in 2008. And we asked, okay, in 2008, looking at all the epitopes doing this comparison, 16 of the B-cell epitopes that are in the IEDB are conserved in that seasonal strain. And that's actually typically pretty representative. We did this for more years, but typically, apparently, like from year to year, there's at least 16 epitopes conserved on the B-cell side. But when we looked at the pandemic strain, it was just 8, which was way less than anything else. So based on that, we would say really the antibody epitopes have changed drastically in this case, and there's much lower number of epitopes that were conserved in the pandemic strain. When we looked at the T cells, though, there wasn't really much of a difference. I mean, this is somewhat lower, but not really that much. And when you then actually, also you could look at the sequence, it became apparent why. Because the main differences of this pandemic strain was that the surface protein had changed drastically in the pandemic strain, versus the internal proteins, which are the preferred targets of CD8 and CD4 responses, were highly conserved and look pretty much like another seasonal influenza strain. So based on that, we hypothesized there would be significant levels of pre-existing immunity on the T-cell level against these pandemic strains. And we tested that. We could synthesize the peptides from the from the pandemic strain, and saw that actually the level of T-cell reactivity in terms of CD4 and CD8 was essentially similar to what the seasonal strain in the previous year was like. And actually, I should have put in some. There's a number of new references that have now come up that have actually tracked also for the outcome of disease based on these conserved epitope reactivity, where there's a Nature Medicine study coming out in England, where they could really show that your level of T cell, the ability of the T cells to recognize the peptides in the new strain is predictive of your likelihood to protect it from severe disease. That's the important part, not from infection. If you don't have the antibody response, you are going to get infected, because the antibody completely blocks the virus essentially at that point. But once it's inside the cell, if you have the T cells, you're going to clear it rather quickly and not get very sick. So that was a prediction. When we made it, it was actually pretty controversial, because everybody wanted to just hear gloom and doom and people dying. Then people didn't. Then, yeah, we could get it published. Okay, but so that was just the motivation, essentially how we can use these tools, like the conservancy tool in this case, to examine data that's in the IEDB and make essentially a new biological conclusion, and make them, by using this, essentially we could, within weeks of the sequence being out, perform experiments that otherwise would have taken a year or 3 to put together. So there's multiple analysis tools available. I talked about the conservancy analysis, population coverage. I asked, given the set of epitopes with no HLA restriction, what is the likelihood of these epitopes to be recognized in populations with different HLA frequencies? The homology mapping tool deals with analyzing structures of an epitope in its source antigen. So if there's, most of the time, I mean, most of the, say, antibody epitopes are linear peptides. This is about mapping them back to the structure if there's one available, or an homology map one. And cluster analysis, which is useful for actually peptides from, say, a set of strains from the same viruses, clusters them together to avoid looking at essentially duplicates. So summarizing this, we have 2 sets of tools. The IEDB prediction tools extrapolate from existing data to identify new epitope candidates that can then be tested experimentally. And the IEDB analysis tools are meant to help examine existing sets of epitopes and gain new knowledge. And with that, I think I'll finish. Like yesterday, I think questions will be best.